for this invitation. I'm very excited to be part of this. As I was hearing the introduction by Professor Newman, I felt so, so much reflected on the topics and on the importance of reproducibility in data science. And I hope I'm going to convince you a little bit on this particular important aspect when it relates to, in particular, fMRI and functional connectomes. So my talk is going to be focused, which is why I made kind of a secondary title, uncovering fingerprints in human brain connectivity. So I borrowed, I hope that's okay, the main focus of the theme because I got very excited about it. Reproducibility in research and in particular in, for instance, neuroimaging, or even more in particular in fMRI is critical. Is critical so that others can reproduce what we did. I will argue is critical also so that we just feel better and secure about the research and the results we are getting, so that we know that it is not just because of a source of variability that we were not able to capture, or because of some issues related to the design or the acquisition of the data or the processing of the data. So I'm going to start. Professor Nima already introduced a little bit on these modalities of MRI. So very briefly, from diffusion weighted imaging, you can obtain information about the diffusion patterns that happen for this particular topic, in particular in the white matter. And on the other side, when subjects are in the scanner, you can also ask them to do particular tasks, particular activities, or also simply what is called resting state, when you ask the subject not to do anything in particular, I will argue, except please don't move the head, because that produces a lot of artifacts in the data. From diffusion weighted imaging, when we combine it with a parcellation of the gray matter, a parcellation of the gray matter, it's a dimensionality reduction approach where we are establishing that our major elements for the study is going to be literally millions of neurons that correspond to certain spatial location along the cortex. So when we combine our favorite parcellation, and there are many parcellations out there, and there are actually groups that are fully dedicated to developing better and better parcellations accounting for different characteristics of the data, we can end up with a connectivity map. What is a connectivity map? It's basically a matrix where you have all the brain regions in the rows and all the brain regions in the columns. And whenever you see a blue dot in this particular matrix, it means that we actually found major fiber tracts through the white matter that are connecting those two regions. So every dot represents the connectivity between two particular regions. This matrix is symmetric because we don't have directionality in human studies about the diffusivity properties. So if A is connected with B, we also assume that B is connected with A. On the other hand, you have subjects doing tasks or actually at rest. And in that case, you have another dimensionality of the data, which is time. And now your y-axis is an indirect measurement based on oxygen replenishment. It's an indirect measurement of neural activity. So the higher is the signal, there is more neural activity that is happening in that particular brain region, and the lower is the signal, the lower is the estimated neural activity, again, as an indirect measurement. So if we apply the same concept of a brain parcellation, we get as many time series here as brain regions we have in our favorite brain atlas or parcellation. If we cross-correlate, if we do, for instance, Pearson correlation coefficients between all the time series, all to all pairwise, we end up with another very interesting object, which is a matrix that we call functional connectivity, which is not reflecting whether there are white matter fiber tracts connecting those brain regions, or at least not directly. What we are doing is we're estimating to what extent it seems to be a functional coupling or communication between those two brain regions for that particular subject when it's doing that particular fMRI task or at rest. So with these matrices, structural connectivity and or functional connectivity, we can make whole brain questions about the subject or a disease progression, or any possible effect induced by a task, learning procedures, etc., etc. We can do it whole brain, or we can still target specific interactions between regions or small brain circuits that you might be interested on, maybe due to the literature or your previous sound results. So if we start focusing more on functional connectivity, there are many ways in which I could order this matrix, which is a whole brain functional connectome. I claim it's a whole brain because we're using brain regions all along the cortex, and we're also including subcortical and cerebellum. A possible way of ordering these matrices is organizing it by what is the so-called resting state networks. These are functional modules or communities, if you want, groups of brain regions that are more coherent. They talk more to each other when subjects are at rest than with respect to the rest of the brain. So for instance, this is an individual subject, individual session of resting state, where we see that the visual is a fairly integrated block in this matrix, which means that most of the visual regions are actually talking to each other. There is functional coupling happening between those regions for this subject as is at rest. We have the somatomotor, dorsal attention, ventral attention, the limbic, frontoparietal, the full moon network, subcortical regions, and cerebellum. It's very important to note that the visual perception of these functional connectomes can be very different depending on how I organize rows and columns. That said, keep in mind that regardless how I organize the rows and the columns, the functional connectome is the same. It's just a matter of representation and how the ordering of the rows and columns is highlighting different aspects of the functional connectome. Okay, in my talk, you have two chances of making me feel old. If you don't know what this game is, this is your very first one. This game, I believe the English term for it was Guess Who? And in this game, basically, you had to ask about characteristics of the subjects, and your mission was to find what is the subject that the other player has as a single card that you obviously cannot see. So, you know, you will ask if is your person wearing a hat. If it says no, then you can discard maybe 1, 2, 3, 4, 5 subjects. And if it says yes, actually it's great because you can discard everybody else and you're reducing the uncertainty of who is the subject that the other player has. Okay, so in this talk, we're kind of going to play this, but we're going to play this with functional connectomes. So the pioneer work by Emily Finn and colleagues in Nature Neuroscience 2015, they actually took data from the Human Connectome project and they started playing this game. Or actually they started putting on the table to what extent with a state-of-the-art acquisition of fMRI data, we can actually play this game. Now, I'm presenting this game more like if you were playing with 2 decks of cards. And the game is as follows. Each card is actually a whole brain functional connectome of 1 session of 1 subject, okay? So how does this game work? I present you 1 functional connectome, and the only information I tell you is this functional connectome is of a person that is also present in this second deck of cards. So you can intuitively think that what you will do is you will look at this functional connectome, the one that I'm presenting to you, and you will start comparing with all the functional connectomes of the second deck. And then whichever seems to be the most similar will be your guess of this must be the same subject. So just in this rationale, there is a lot going on about our expectations. We actually expect that when you take 1 subject for a second time in the scanner and perform the same task, let me add within a very short period of time, maybe hours or days or no more than weeks, you have great expectations that the functional connectome of that subject, the second time you acquire the same task, should be much more similar to the very first time you acquire it on that subject than on any other subject. And this is our intuition, and this speaks of our aiming to have reproducible data. And reproducible data in this case will be related to the sequence, the fMRI scanner, the settings, the specific instructions that are given to the subjects, and how we do the post-processing of the data until we obtain the individual functional connectomes. So there are all these ingredients that play, and how committed is the subject to actually follow the instructions the 2 times if you want. So a mixture of everything I have said and a few things that I'm not even mentioning makes an impact on whether it's true that the functional connectome of a retest or day 2 of a subject doing a task is more similar to the first time the same subject did it than to other subjects doing the same task. So in this particular game, you will play all the cards. So I will show you 1 functional connectome at a time, and then you look at the second deck and you try to match with the one that looks the most similar. Identification rate, which is the y-axis in all these figures, is the percentage of times that you were right, that you were able to match the functional connectome day one or test of subject X with the functional connectome day two or retest of that same subject. The more times we are able to do this, it speaks better about the data. Data in a very wide way, data including the fMRI settings, the acquisition sequences, the post-processing, and the experiment design and instructions to the subject. It affects, it speaks well of all these joint experiments, not just of the data per se. So we can see that when we read all in this figure, it means whole brain functional connectomes comparisons. So identification rate seems to work really well at the whole brain level when you compare resting test and resting retest. You are comparing the same fMRI condition 2 times. It works, it's more challenging, although it kind of works and it has identification rates fairly high when you do, for instance, when you compare functional connectomes that come from rest with functional connectomes that come from an emotion task. And it works also fairly well when you compare a working memory task with respect to resting. And please keep in mind that when you add the complexity of different fMRI conditions into this game, it's obviously more challenging because we have expectations that you undergo a functional reconfiguration when you're doing a task with respect when you're doing a rest, which is actually a topic that we are very much focused on in the last year in the lab. So the big picture is when you see 1 to 8 is actually the so-called resting state networks. And when you focus only on those resting state networks, how you see that the identification rate, which is the level of fingerprint that you see in the data, if you wish, it varies across resting state networks. So I haven't told you yet one very important aspect of this game, which is when I take one functional connectome and I compare it one at a time with all the others to decide or to predict who is the subject, I haven't told you how we do it. So let's get deeper into this. So imagine you take one functional connectome which remember is a symmetric functional connectivity correlation matrix, is the Pearson cross correlations pairwise of all the fMRI time series, ball time series. So imagine I give you one functional connectome, and we're playing this game with a small cohort, so for the ease of simplifying the game. So you take one subject, and I ask you, okay, you have to figure out who is this subject. It can be subject 1, subject 2, all the way to subject 20. And you compare one at a time the functional connectome with the functional connectomes of the retest pool, or deck 2, or day 2, as you wish to call it. So what we're going to do is simply the Pearson correlation coefficient of the entire connectivity profile. So in this case, the question mark subject, when we compare it with subject 4, we actually get a very nice linear cloud, which actually is reflected by a very high correlation coefficient. So that means that it really looks like it's this subject, but you can still keep checking on other subjects. And for instance, the correlation with this other subject, with subject 15, is actually really bad. So that means that it's very unlikely the correlation is barely above 0.4. So that actually means that given this profile of correlations, comparisons of your question mark functional connectome with all the other functional connectomes, your bet, your guess, your prediction will be this must be subject 4. Because the retest of subject 4 really looks very much like the test of this question mark subject. Now, based on identification rate and how it works, if this was correct, if it was subject 4, you get a +1 if you want, you get a 1 more successful event. And then the ratio between successful events and the total number of attempts is actual identification rate, which is why it's constrained between 0 and 1. Now, this is great, but there are 2 aspects that is important to consider on identification rate. One is that it's an all or none score. What do I mean by that? Okay, let's assume that unfortunately it was not subject 4. So let's assume that based on these comparisons, based on correlation coefficients, subject 4 retest is the most similar to the question mark subject test, but I'm telling you that unfortunately it was not the subject. From the standpoint of identification rate, now it didn't matter if in reality it was subject 12 or subject 15. But if we are a little bit flexible on the idea of a fingerprint, it's a much better situation if the subject was 12 than if he was 15. Because if he was 12, basically I just needed one more shot and I would have figured out who was the subject. If he was 15, it basically means that I would have needed 20 attempts, excuse me, 20 attempts actually to name each and all the subjects until I will figure out the subject. So at this situation where it was 12, we consider there is a much better situation with respect to the fingerprints in the data than a situation where it was IF 15. Whereas identification rate, you are either the most similar or it doesn't matter, you get a 0 otherwise. The other part that is interesting on identification rate is that it's a little bit difficult to compare identification rates between data sets. And the reason is that the null model, or if you wish, the null chance of making the right choice in identification rate is very related to the cohort size. You can intuitively think in this game that if I have 5 subjects, my chances, my options that just figuring out the subjects by chance are way higher than if I have 100 or 1,000 subjects. So this is also something to keep in mind when you're using identification rate, that the basal chance, it's very much dependent on the cohort size. Okay, so then thinking about all this, and this is work led by Dr. Enrico Amico, former postdoc in my lab, and now he's in EPFL in Lausanne as a PI. We said, well, at the end of the day, when you take one functional connectome and you compare one at a time with all the other functional connectomes, you're doing all possible comparisons. Why don't we put this into a matrix? And then we did something really simple. From the functional connectomes, 2 functional connectomes per subject, that's denoted by FC1 test, FC1 retest, then it should be FC subject 2 test, FC subject 2 retest, all the way to FCN retest. When we put the comparisons into a matrix, don't get confused, this is brain regions by brain regions, these are functional connectomes, but this is subjects by subjects. So this is what we call the identifiability matrix. What is the identifiability matrix? It's simply the correlation coefficient of the entire connectivity profiles of the functional connectomes of all subjects test in the rows versus all subjects retest in the columns. Now, the only thing that matters is that you order the same, the subjects in the rows and the subjects in the columns. It doesn't really matter the order, at least for now, but it's very important that you do it in the same way. Why? Because then the main diagonal gets a very important meaning. The main diagonal is for each subject, how similar is the functional connectome test versus retest. Off diagonal is comparisons of different subjects, test versus retest, all to all. So the idea of reproducibility, or the idea of fingerprints in this particular experiment, is reflected on the idea that we would like that the main diagonal is brighter than the off diagonal. Because the main diagonal being brighter than the off diagonal means that the functional connectons of the same subject looks alike more than between different subjects. Now, we wanted to put a score to this intuitive idea. So the way we do it is actually very simple. For every entry, if the entry belongs to the main diagonal, is reflecting how similar is you with yourself. So the average of that, the average of the main diagonal, we call it I self, which is denoted here as the average of the main diagonal. And we want that to be high. But what does it mean to be high? It only means anything if it's relative to how similar are different subjects. So basically we want a contrast. The identifiability matrix is good when there is a contrast between the main diagonal and the off diagonal. So we want that the mean of the main diagonal is higher than the mean of the off diagonal, which we call I others, or on average, how similar are functional connectons of different subjects in your data set. So this gives you a value. It's a score that we multiply by 100 just for simplification purposes. So let's develop some intuition on how this works. I'm going to present you for a cohort of 20 subjects, all doing resting state, 3 different scenarios of identifiability matrices. So in the first one, everybody looks the same. Everybody's functional connectome correlates 0.8 or 0.9 or even more with everybody else, which corresponds to a very low identifiability score. Differential identifiability score is how we call it. On the other side, not all functional connectomes are the same. Actually, they are fairly different, but unfortunately, even the main diagonal, they are not very similar. So it's kind of the opposite problem of this one. The functional connectors look different, I mean, they are different people doing the same task. There should be some variability, but even when it's the same subject, it's not very prominent that there is a fingerprint. So this is a better score than this, it's 16 points, but still this is not very ideal. So the problem in the first one is that I self, in a high similarity environment where all the functional connectors look pretty much the same, I self is very similar to I others. Excuse me. On the example on the right, it's kind of the opposite problem. In a low similarity environment where all functional connections look fairly different, it's also true, unfortunately, that I self looks similar to I others. In the middle one is where we would like to be. Let's put on hold this subject, and I will mention on it in a minute. The tendency is that the diagonal is brighter than the off diagonal for each and all the rows and columns. So I self tend to be greater than I others. So this is your second chance to make me feel old if you have never seen this movie, but this is a movie where everybody looks like John Malkovich. So when we have a data set like this, we say that it has the John Malkovich syndrome. In this data set, functional connectons, it doesn't matter if you call the same subject again, or if you recruit different subjects, because all the functional connectons look pretty much the same. We call this one the carnival syndrome. Everybody's wearing a mask, but if you recruit the same subject again, it's wearing a different mask. So we don't know who are the subjects. Here we don't know who are the subjects because they all look the same. Here we don't know who are the subjects because they keep looking differently. And this is where you want to be. You want to be in a situation with your fMRI task design and acquisition and instructions and processing, where when the subjects come again, they look closer to themselves in test or day one than to any other subject. Excuse me. So this is where we want to be. And then I'm going to present you in the rest of the talk, basically a method to try to uncover in a data-driven fashion the fingerprints that are present of your data. Basically a way maybe to move from this scenario, your data, to this other scenario. But honestly, even if you don't get convinced by my method, I hope you do, but even if you don't at the end of the talk, the most important slide of my talk today is that if you have EEG, MEG, fMRI data, try to do the identifiability matrix. Try to look at this because you're going to learn so much about your data, as I can claim, for instance, that 2019, 18, 16, 17, 15, the test session of the subject 15 in this data set, there is something going on. Because it's not only that it doesn't look much like itself in the second day, it looks way different to everybody else in the population. Just looking at this matrix is so easy to capture this type of things, and then you might want to dig what was the head motion, whether you did the regression right or not, whether the subject maybe it didn't understand the instructions the first time, who knows, it can be a number of things. So even if you don't like, I hope you do, but even if you don't like all the methods I'm going to present you after this is the most important slide of the day. If you want reproducibility and fingerprints in your data, you have to look at these matrices. Because if your data is here in the John Malkovich syndrome or is here in the Carnival syndrome, you can try to make connections with depression scores, with disease progression, with working memory capabilities, with executive function abilities. And you have data so that you can do it. And there are many frameworks out there very interesting to do those mappings. But your data is telling you that you are not in a good situation to do this. Here, because it all looks the same. Here, because you cannot rely on a single session of the subject, because when the subject came again, the functional connectivity estimations were completely different. So this is the most important part. And now let's see what we did after this. So we said, okay, it's nice to provide an alternative to identification rate that takes fingerprints a little bit more like a continuum, where it doesn't have to be the most similar. We're actually measuring to what extent your retest is more similar than the rest of the population. But we wanted to do more because imagine your data is here. Okay, then what do? You have spent all this money, you have made this experiment, you put so much effort, you recruited subjects, and then you got a relatively low fingerprint score. Well, we wanted to give you a last chance because the data is there, the estimations are there, and we thought that there were things that we could uncover a posteriori. By a posteriori, I mean after we have fully processed the data and obtained single session individual whole brain functional connectons. So basically, and here is my metaphor, it was a very difficult exercise because we wanted to clean your data. And the situation is as follows. Imagine I open a garage door and there are 50 bikes there, except the 50 bikes are full of dirt, are really dirty, are full of dirt and mud. And imagine we have, and I'll present you in a couple of slides, a very powerful cleaner. But the problem is that we have never seen a perfectly clean, brand new, shiny bike. We kind of know how a bike looks like. We kind of know how a functional connectome looks like. But even the concept of what does it mean a zero noise functional connectome is almost ill-defined because we will never see one. We will never see a noise-free functional connectome. But we kind of know how a functional connectome looks like. So we have this very powerful cleaner that I haven't introduced you yet. We have the bikes, the functional connectors that we want to clean, except if we do an overdose of the cleaner, we're going to melt parts of the bike. So how do we know what is the optimal dose that we will be using to clean the functional connectivity data? Well, we're gonna let the identifiability matrix and the differential identifiability score, mean of the main diagonal minus mean of the off diagonal, to tell us what's the right dose. So what is the cleaner? It's very, very simple and very intuitive. It's called principal component analysis. The only thing, the particular thing is that the input for the principal component analysis that we add, it has 2 full connectivity profiles per subject, because remember we have test and the retest. We vectorize, we take the upper triangular because remember these functional connectors are symmetric because they come from correlation coefficients. We vectorize one at a time, subject 1 test, subject 1 retest, subject 2 test, subject 2 retest, and so on. To tell you the truth, the order of the columns, it doesn't matter, but it's just for our own understanding and having a good, clear index of who is who, that we do test, retest, test, retest. But remember, for PCA, it doesn't matter what's the order of the columns. What do we get from principal component analysis? We get the principal components, and we get, which are linear combinations of the entries, and we get a method that it's deterministic, and it explains all the variance. What do I mean by that? If I use all the principal components to reconstruct the data, I get a perfect full reconstruction of the data. Now, in our metaphor, that means we haven't used our dissolvent at all. So what is going to be our dissolvent? Our dissolvent is going to consist of putting all the functional connectors of your cohort test, retest on a principal component analysis framework. And then we're going to start considering what if we drop the last principal component and reconstruct the data? How well are we doing in our identifiability matrix and in the differential identifiability score? Are we doing better? Are we cleaning the bikes? Yes, okay. Then let's try to remove the last 2 principal components. By last and last 2, excuse me, we rank them in descending order of explained variance, because our hypothesis is that there are 3 kinds of principal components in functional connectivity data at the cohort level. The first kind is global properties, just a human being doing that task. So that might be represented by the first or the very first 2 components. Then there are components that carry information about the subjects, the ones that carry the fingerprints. And then there are components that are dependent on things that have nothing to do with the fingerprints. What we don't know and we didn't know, and is data specific, is what is the frontier between the second and the third type. Should I drop the last 10 components, or the last 27, or the last 32? So, okay, with all this in place, we made a, excuse me, we made a very small experiment where with 20 subjects doing rest and having test retest sessions, we check if this works. And again, one at a time, we were removing the last component with the least explained variance, the last 2, the last 3, and so on. Okay, so this is the experiment. And the way to read this is when we are on the right of the center plot, we haven't done we haven't used the dissolvent at all. We have not removed any principal component. It's like decomposing into principal components and reconstructing the data with all the principal components. Now, if I spray a little bit of the dissolvent, we are removing the last principal component. We produce the identifiability matrix with it. By the way, this is the identifiability matrix at the starting point when we have not used the dissolvent by using a state-of-the-art fMRI processing method. So it has 16 points, which if you remember is the case of the right on the 3 examples that I showed you earlier. If we spray a little bit of the dissolvent, we drop the last component, we reconstruct, we see how we're doing, and we get a little bit better differential identifiability. So we keep going, we keep going, we keep going. We're spraying more and more on the bikes, the functional connectomes. And at some point, we actually get to a point where we have more than 28 points of differential identifiability. So we're doing much better than at the beginning. Here is how we were doing at the beginning on the identifiability matrix. This is how we're doing at the optimal level of the composition. Why I say it's the optimal level? Because when we keep removing components and reconstructing, after removing more than, this must be 21 components, we're actually spoiling back the fingerprints. We are cleaning, we are dissolving parts of the bikes that made the bikes to look different, rather than doing any cleaning of the dirt and the mud that is present on the data. Actually, if I keep overdoing it, I can even reach regimes of differential identifiability that are even worse than my baseline, which is what I was at the beginning. So this is the full spectrum on the principal component, the composition and subsequent reconstruction. As I like to read these plots right to left, as I'm cleaning the data more and more and more and more. So more cleaning doesn't mean better necessarily, and thanks to the differential identifiability framework, we can find the optimal level.
Joaqui?
Yes, please.
Sorry, your voice has become much lower than before.
Oh, I'm sorry about that.
Yeah, it's better now.
It's better now. So as I was saying, thanks for letting me know. These plots is better to read them right to left. This is the full cleaning profile of the data, and more cleaning is not necessarily better. And we let the differential identifiability framework find the optimal dose of the cleaning, which in other words means how many principal components to remove from the data and then reconstruct. And remember that when we reconstruct, we get back clean functional connectomes that uncover the best of the possible fingerprints that are present in your data. So, okay, this was a small test with 20 subjects doing resting state. So we aim bigger, and at the time we took the official list of 100 unrelated subjects in the Human Connectome project. I can tell you nowadays that we have reproduced this with an enlarged list of is somewhere close to 440 subjects, and the results hold. And we took all the fMRI conditions that are present in the data, which include resting state, but also 7 different tasks: emotion, gambling, language, motor, relational, social, and working memory. Now, note that the duration, the number of frames acquired on every fMRI task was very different. Okay, so the exciting part of this figure is that we can say that this method is universal to the fMRI task, at least to the best we can do, which is using all the fMRI conditions available. The method always works in the sense that it's always worth it to use it. It always finds an optimal level of reconstruction for which the differential identifiability is higher than the baseline. You can see that it tends to peak in the middle. Just to make a note, I said that it's 100 subjects. You might have expected that this would go up to 200 because we have test retest, so the dimensionality of the data is 200. Just trying to be conservative, what we did is a bootstrap method in which we ran several runs of this by taking only 80% of the subjects, which is why the dimensionality goes up to 160 only. The problem, the good problem, is that the standard deviation is so tiny that it gets inside the circle, because it really doesn't matter. Differential identifiability and optimal reconstruction is almost impossible by how it's conceptualized, that is going to be driven by a few subjects. But just in case and to prove it, we decided to do it that way. You will notice that the peak tends to be in the middle, which is more or less peaking at half the number of components. So it looks like a good procedure to use half of the components in this data, in this type of approaches. There are different reasons for this. One is that half of the number of components matches the number of different subjects in the data. So it kind of makes sense to think that this is a good approach, that it will give you, if not the most optimum, at least a very good level of differential identifiability and a good cut point to actually reconstruct the data. As you make the cohort larger and larger, this is not necessarily the case. And I don't have a figure here, but as your cohort is above 400, for instance, the peak, the optimal peak on the number of components tends to shift to the left, which means you are needing less components than the number of subjects to actually uncover fingerprints in the data. So other than that, there are certain tasks for which the level of fingerprints is very high, like for instance, in the language task. There are others where it's not so high, like for instance, in the motion task. And just to be fair, the comparison with REST is complicated because all these tasks are 2 to 5 minutes, and the resting state sessions were, I can't remember if it was 12 or 14 minutes. Anyway, more than twice or sometimes 3 times longer than the fMRI task. So I'm just saying it's a little bit difficult to put REST in the context of all these tasks because we're having more points, and I have a figure dedicated to that later. So for the sake of time, I'm going to skip a few things. I hope it's okay because I want to focus on a few things. So one thing we were said is, okay, this framework is great, but it's not like everybody has test retest. It's not like everybody calls the same subject twice. By the way, I'm setting the slides. So the slides that I'm skipping, if you have questions afterwards, just reach me by email and I will be very happy to address them. So we were said not everybody has test retest. This is very expensive, very complicated, and most of the studies are actually cross-sectional. So we said, okay, yes. So maybe what we can check is if your acquisition is long enough, you could actually split a single acquisition into test and retest by literally creating the first half and the second half, mimicking a test retest situation and running this framework. And this is what we did for the 4 resting state sessions that are present in HCP. And it actually works, and actually it works great. And it actually makes sense because a continuous acquisition than a posteriori you split into 2 halves, you could argue is the most ideal test retest scenario. The subject is not even getting up from the scanner. The subject is not being interrupted by anything. So this is the most ideal situation of a test retest. So as kind of expected, we reached unprecedented levels of differential identifiability up to 40 points. Keep in mind that how to read the points, 40 points means that in your functional connectivity data on average, the test retest of the same subject correlates 0.4 more than test or retest of different subjects. So that is a very high level of fingerprint.
Wenn du möchtest, es ist aktuell Data Science Week, das ist organisiert von
So,
und dann Labor.
Exploiting that.
Please put yourselves.
Ja, es geht im Generellen
Clemens?
Bildgebungs.
Clemens, your mic is on. Okay, sorry. Yeah,
no worries.
Please.
No worries. So exploiting that in resting state, we have a very large acquisition. We try to check what happens when what is the effect of a scanning length on our framework. In other words, can we use our framework regardless what's the scanning length, or it only works when it's longer than a certain number of minutes, or how it behaves. So basically in these plots, you have the number of fMRI volumes, 1200 is the total when you're using the entire resting state and scanning length. And then as we move to the left, we are shortening the time series, obtaining the corresponding functional connectomes, running the differential identifiability framework, and getting the differential identifiability score when you don't use the principal component, the decomposition reconstruction, in red, and when you actually use the optimal level of reconstruction. So this figure, for me, it's really, really exciting because before running this experiment, if I were like a salesman, I wanted to be able to tell you that with our product, we make your resting state, let me say, we make your resting state 5 minutes acquisition as good in terms of fingerprint as if you will collect 9 minutes, for instance. I wanted to be able to say something like that. We can say much more than that actually, and I was very impressed with this. Give us 200 fMRI volumes in resting state, or if you're not convinced, let me say 300, and you reach fingerprint levels, scores of differential identifiability that you will never reach even if you go beyond 14 minutes of acquisition. If you agree with me that here things are starting to saturate, and I don't expect any big bump of differential identifiability or any other measurement of fingerprints after this scanning length. So that's a very strong message because it means that our method is not just compensating for a relatively modest number of time points to compute the correlations for the functional connectomes, is actually cleaning the data in other ways that simply extending the scanning length cannot do. And the second point that is very important is that it looks like to use our method is worth it regardless the scanning length. Even for 100 or 50 volumes, even, okay, for 50 volumes, you just shouldn't do any study. But if you do, it's still able to uncover more fingerprints than when you don't do our method. So it's really worth it to use it regardless the number of fMRI volumes that you have. And it not only compensates for relatively short scans, it actually uncovers fingerprints that cannot be compensated by scanning longer and longer and longer. So this was a very important message in our study. Okay, so after this, we had so much fun with this work that we said, can we complicate ourselves a little bit more? And I'm gonna dedicate 5 minutes, maybe 6, to how we complicated ourselves more. And there are things that I'm not going to cover, but I'm more than happy if a posteriori, as you check on the slides, you have any questions for me. So the most important one that I want to present you is the worst nightmare for any fMRI engineer. Let's collect data in different sites, which is actually very exciting. That was mentioned as one of the motivating areas of research in the ICN DataWeek. So we had, for a very small cohort of subjects, 2 pairs of test retests per site for site 1 and 2 pairs of test retests for site number 2. So that means that we had 8 functional connectomes in total per subject. So that allowed us to expand the concept of differential identifiability framework to a multi-site scenario. And basically what we wanted to know is this automated data-driven cleaning based on principal components, so something that will uncover fingerprints when the test and retest are from different sites. So now you can think for 2 sites that we are expanding the matrix by 4, because this is what we used to have before, subjects by 5 subjects, test, retest, site, 1 site. Now I have the same sub matrix, let me call it, for the second site. And this is the exciting part. Now I have when the test is collected in site 1 and the retest in site 2, and when the test is collected in site 2 and the retest in site 1. So I'm going to briefly skip the math, although the rationale is the same, mean of the main diagonal minus mean of the off diagonal. So you can think that I can track 4 differential identifiability patterns, the one within site 1, the one within site 2, and the really challenging ones, the one when test is collected in site 1 and retest in site 2, and the other one when test is collected in site 2 and retest in site 1. And keep in mind that it's not just 2 different imaging sites, the model of the scanner was different and the sequences were slightly different. So these are the 4 patterns of differential identifiability when we put all the data together. So there is a test retest of site 1 and a test retest of site 2 per scanner. Sorry, per subject. So as we know, the within site, it actually works. And we were able to uncover a very high level of fingerprints. In light blue or cyan, you have site 1, and in purple, you have site 2. We kind of expected this. It works. It made us happy, but it was nothing out of the ordinary given our former results. These are the ones that are really interesting. When you do inter-scanner site 1 versus site 2 test retest, or site 2 versus site 1 test retest, there are many more components that you cannot take, that you have to clean. You cannot access many components that are probably contaminated with effects of one of the scanners or the other. But when you actually get the optimal level of reconstruction, you actually reach the level of differential identifiability that you have in your data within site when you don't use our method. So in summary, what this means is that using differential identifiability across scanners test retest for functional connectomes, from the standpoint of fingerprints, and I won't go beyond that, but for the standpoint of fingerprints, it fully compensates the multi-site effect, because you get the level of fingerprints that you get when you don't use our method and you collect the data in the same scanner. And for those that are into the fMRI world and processing, this actually works when you don't do global signal regression and when you use global signal regression. And this is the average of the behavior of the 4 patterns within site 1, within site 2, site 1, site 2, site 2, site 1, how it behaves in terms of differential identifiability. So This is the really exciting result, the differential identifiability in the original data processed with the state-of-the-art methods. Look at these secondary diagonals when you actually reconstruct the data, how they get much more brighter, and you can actually reconcile that for most of the cases, not all, but for most of the cases, this is the same subject collected in different sites, and how that was barely possible to do in the original data. This is with no global signal regression, with global signal regression. In the training set, when we literally the PCA and the reconstruction on the same data, and in the validation set where we freeze the components, we freeze the linear combinations as the dimensions for the data, and we impose the reconstruction on runs of the same subjects that were not used to actually obtain the principal components. And it actually works. So this speaks for validation, for the scanning length, as I mentioned before, for multi-site studies, and for new runs of the same subjects. Unfortunately, as of today, I don't have anything that could convince us that we can use the results for future subjects, but it works for new runs of the same subjects. Okay, very, very quickly, I just want to share that we're extending also this approach to the concept of twins. And very quickly, and this is a manuscript in preparation that is going to be on archive by next week, I can actually share that when you look at the structural connectomes and functional connectomes, we see the expected gradient of fingerprints. Test-retest of the same subject is always higher. When the retest is not you anymore, but your monozygotic twin, which share 100% of the genetics and in principle, a lot of environment, it's lower than the retest of the subject, but there is a very high fingerprint, and the gradient keeps lowering when your retest is your dizygotic twin, between 40 and 60% of the genetic material shared and a high shared environment, most likely. The level of fingerprint is smaller, but still way higher than a null model where you randomly match the subject. So the exciting part of this is that actually this framework also allows us to see fingerprints in twin studies. And for the sake of time, I'm going to stop here, but basically we have also tested that you can run these measurements not only on functional connectivity data, but on network measurements derived from the functional connectivity data. And last but not least, this is our website, and I said in this because you can also check all the ongoing work on fingerprints that we're doing. Part of it has been presented here. And if you are interested in using it with your data or in checking our framework, in most of the papers, we actually share a small MATLAB package with the code and with some, excuse me, a small data set so that you can run the entire thing. And it should be very helpful so that you eventually can run our frameworks with your own data. And I wanted to thank you for your attention. If you're interested in collaborating with us, maybe visiting us or working with us, please send me an email. And I'd like to thank again Newman and Safa for inviting me for this great IC and Data Week. And I'm happy to answer any questions.
Thank you so much, Joaquin. Thank you. That was a very, very interesting and promising talk. Thank you. Timon has a question. Do you wish to read it, or shall I? Timon, do you want to step? Shall I read it? How subject individual PC characterized. Do you agree that this could be helpful? WRT data cleaning for across patient analysis, Idit being small.
For across what analysis? Sorry, I missed it. So the question is whether you think with respect to data cleaning for cross-patient analysis, whether the within-patient PC could be helpful.
So, okay, it's a very interesting question. When you have patients and you have not a cross-sectional but a follow-up, the framework is very interesting, but one has to be very careful that you are not unintentionally removing effects of the disease. This is one part that is very critical, because there are actually papers appearing where something that will be very equivalent to the eye self-measurement, which is how similar is my functional connectome in day 1 versus day 2, is actually related to the disease. For instance, there was a very interesting paper in 2018 where they did that in a cohort with schizophrenia. And the test-retest measurements in patients with schizophrenia are more different than with respect to controls. So in that case, I will argue that you don't want to remove that effect because it's part of the disease that the consistency in the functional connectome is different. But rather than that, you can actually learn a lot from the identifiability matrix when you look at cohorts that include patients, including whether there are modules or communities in the identifiability matrix, which means there are subjects that look more alike others than other subsets of subjects. Something that we have also seen when you do certain tasks that are highly engaging, we see that the functional connectomes of the different subjects tend to look more alike than when they do more low engaging tasks or even resting state. Did I answer the question more or less?
Okay, yes, thanks. So I have a question. You've been talking from...
We cannot hear you, Julian.
Still not?
We hear you a little bit low voice, low volume.
Okay, can you hear me better now?
Yeah, I think so.
Yeah. Okay, so my question is, how do you make sure that within your lab, all these different data sets that have patient IDs or subject IDs, and some analyses are cross subjects, some analyses are within subjects, how do you manage that all your lab members are actually on the top of what is where, and how do you manage all the data? Because that is a lot of gigabytes and terabytes of raw data. Like, how did you manage to get this all organized in a way that it is easy and effortless for you to work?
Okay, so the way we do it is there are a couple of members of the lab, well, one being me, that take care of the processing part. And we have, I don't know if to call it a data center, it's basically a machine with literally 20 hard drives where we have all the files and the process files. Now, for people doing projects that are, their starting point is maybe the functional connectomes, or maybe one step before the ball, the femur right time series, we actually share with them only that part so that they don't get their computing workstations completely overloaded with intermediate steps that are not needed. Yeah, I think this is mainly the way we do it. And then,
so you
know, the data is anonymized and
team roles are most important.
Can you say that again? I'm sorry.
Would you say that the individual roles, having roles for certain researchers that they take care of processing and managing it, is the most important aspect of that? Or what is the most important aspect?
So the most important aspect, I will say something very obvious, but the most important aspect is that you don't only process your data with the best possible state-of-the-art methods that you're aware of, but that you look at your data. Looking at your data will include, in this case, looking at the identifiability matrices, but not only that, looking at every individual functional connectome that you produce so that you're able to detect any possible problems that might be happening.
Okay, that's great. That's interesting. Victoria is also having another question. Victoria, do you want to speak for yourself?
Okay. Hi, Joaquin.
Hello,
Thank you very much for your talk. Really, really interesting. So I will have a lot of questions, but I will go to one of those. Okay. Actually, for the last part of your talk, you were referring about this between sites problem.
Yes.
I think, maybe I'm wrong, but the idea of that PCA works similarly when you took actually the half of the components. I mean, and when I say similarly, is that from the inter-site relationships, it looks like the variance that you are taking aside was the variance that was related to the between sites problem itself. I think that the noise, because yes, you were taking with different scanners, I believe. So absolutely, what do you, have you ever think, I think you might do it, but it seems like you can also tackle this problem by using transfer learning approaches, because it is actually a change in the domain, the distribution of what you're using from measuring the correlation coefficients have changed a little bit. It's similar, but have changed a little bit. So maybe by transfer learning approaches also could we tackle this?
I'm sorry, I missed. Did you say transfer learning?
Yeah, transfer learning is.
Can you tell me in a sentence or two what is it? Unfortunately, I don't know what is transfer learning.
Okay, so the idea is that you have, because you have two different domains here in a way. So your data is similar, but a little bit different because the distribution has changed a little bit because you have changed the tools in which you are making the measurements. So by doing transfer learning, whatever you have learned from one side in this case can be also be like, you can use it for tackle a new problem that is similar, you know, in a way, but it's a little bit different. So like in that sense, you may have some tools that help you to make the distribution similar.
I see. So because I never knew about this term, I'm sorry, transfer learning. I'm not sure if it will fall into this category, but an ongoing project that we have that unfortunately I didn't have time to show anything. We are trying to tackle this problem from the point of view of projections of data from one scanner to the other. I don't know if that could fall into the category of transfer learning.
Yeah, that's similarly, like by doing transfer learning, you may use like you try to match the distribution between these two news sites. So in that sense, you may use the same PCA algorithm between the program.
Yes, because the part that we're excited about this part of the projections of the data, we use it through something that is called manifolds, and we still don't have enough results so that I'm comfortable sharing, but it looks very promising. The exciting part of that avenue is that for what I presented here, I'm very comfortable saying we have resolved the problem for the subjects that you already have in your cohort. Unfortunately, it's much tougher to give you something that you could automatically apply for new subjects that you acquired. The part that we are working on the projections looks promising for being able actually to give you something that you simply apply to new subjects, and it should work to project them to one scanner or the other, or a neutral spot in between, basically.
Yeah.
Okay, great. Thanks for the interesting discussion. Great for talk, Joaquin. Really nice.
So much.
Move on further